I'm just going to talk about a project that he's a big part of, the Genomic Encyclopedia of Bacteria and Archaea.
Thank you, Dan. And conveniently, I'm going to lead off with mentioning Dovzhansky because that is the driver behind pretty much everything I do in genomic studies, is trying to make use of evolutionary information or to use evolutionary analyses to inform genomic studies. And this is particularly important this year in the 200th anniversary of the birth of Chucky D, as we like to call him. And there have been many worldwide celebrations in Darwin's honor, and there are many more, and I like to think that this Genomic Encyclopedia project fits in pretty well with this 200th anniversary of Darwin's birth. So in terms of microbes, the guiding light of evolution of microbes has been the ribosomal RNA tree of life that has provided us with the phylogenetic perspective for bacteria, archaea, microbial eukaryotes, and life in general that was missing for a long time. Unfortunately, the tree is not very happy with we genomicists in a variety of ways, but in particular, the way that I want to talk about is something that is sort of obvious to a lot of people when they look at the genomes that have been sequenced and ask where they fit in the tree of life. So if you go to the Genomes Online database, which has this really nice tracking system of available or in progress genomes, you can see this amazing increase in the number of bacterial and archaeal genomes that are available over time. However, these genomes have not been sampled evenly from across the tree. So if you look at the phylogenetic tree of bacteria, and I'm using basically parasitizing some figures from a really great paper by Phil Hugenholtz that he wrote in 2002 that basically pointed out this problem when doing a summary of all the genomes that were available. So if you look at the 40 or so major lineages of bacteria that were known at the time, the phyla, the genome sequences that were available came mostly from 3 of those phyla. So the Firmicutes, like the ones Ashley just talked about, the Proteobacteria, and the Actinobacteria, which includes things like Mycobacterium, Streptomyces. That's where most of the genomes were and still are from in the tree of life. Some other phyla were poorly sampled at the time, but there were at least one genome or a couple of genomes from those phyla. And the same general trend was seen in archaea, although I'm not showing that on this slide. And so Phil pointed out, and many other people had pointed out, that tree-based guidance for sequencing genomes would be really beneficial for many reasons. This has been done in many animal sequencing projects, in particular many of them led by the JGI and by the chair of our session, has done many of these genomes where they're sampling from across the diversity of animals. There have been many small projects in bacteria and archaea to fill in the tree, but no sort of big-scale projects to really systematically fill in the tree. And so while Phil's paper, or right after his paper came out, or while it was coming out, we were working on a proposal to the NSF Tree of Life program when I was at Tiger to fill in some gaps in the Tree of Life, and then Phil's paper came out, so we basically used everything from his paper to justify our proposal. And we got funded to sequence the first genomes from 8 phyla of bacteria for which there were cultured species but no genomes available. And we had sort of these 3 driving forces behind this project when I was at Tiger, which was we thought more genomes would help us resolve the phylogeny of the bacteria, the overall phylogenetic picture. We thought it would both launch biological studies of these phyla and lead to discovery of novelty. And we thought we could leverage this data for some sort of environmental studies of organisms, although we weren't really thinking about metagenomics per se at the time. Just as an example, we just had a paper on one of these genomes come out a few months ago in a collaboration with Don Bryant and a few others on Thermomicrobium roseum. And it certainly is true that when you have the genome of a novel organism, in this case, it had been de-phylaized since we started the project, but it was still phylogenetically novel. You can learn lots of things about the organism, as everybody knows. You can learn about processes that you may not have found in any other genome previously sequenced by sequencing these novel organisms, and inform a variety of things. And We think we learned a lot from the course of this NSF Tree of Life project, and we're still writing up papers related to this project, but the tree is still kind of pissed off at us. And the reason for that is actually pretty simple, and I think we didn't really think about this that clearly when we did this NSF Tree of Life project, which is the diversity within phyla of bacteria is immense. Some have hundreds of major lineages that you might call orders or at least families. And even when there's a group that's been well sampled, like the Actinobacteria, very few of these different lineages have been sampled. So here's, I know you can't see all the text here. Here's a list of the orders and families in the Actinobacteria, and most of the genomes came from only a couple of these. So within phyla, the sampling was really poor, and in retrospect, we should have thought that, so for example, resolving the phylogenetic relationships among the deep branches in the bacterial tree, one genome from 2.5 billion years of evolution, not that helpful. I mean, it was cool and everything, but it's just not the level of resolution that you need. So while we were sort of, kvetching about our project, I moved to Davis and I started to interact with JGI, and they were in the progress of some discussions with a variety of people, such as Rick Stevens and a group that was suggesting sequencing all the Bacteria and Archaea in Bergey's manual, for example. And there were a variety of other discussions going around at JGI and other places that led us to have some workshops and some meetings where we said, we really need to fill in the tree in a much better detail than had been done by any of the projects previously. And basically the idea was to use the tree not just to pick individual representatives of phyla, but to really guide the sequencing of genomes. And so we started at JGI this pilot project that we called the Genomic Encyclopedia of Bacteria and Archaea, that basically the goal of this project was to do a better sampling from across the bacterial and archaeal tree, and to then test how beneficial this sampling would be for a variety of purposes in studying bacterial and archaeal genomes. So the way it worked was we selected about 200 organisms using the tree as a guide, and I'll get to that in a little more detail in a minute. We developed a sort of pipeline for getting DNA from all these organisms, and the goal was to sequence and finish 100 of these genomes, and then to release the data to the community for everybody to use with no restrictions. This is not to guide, individual studies of these organisms, but to benefit everybody in the community that wants to use these data. And then our task for the last year or so has been to try and assess the benefits that come from this type of project in order to determine if it's worthwhile to sample further after this pilot project. So the way it has worked has been to take advantage of some of the really great people at JGI who happen to be very well positioned for helping us select genomes. So Nikos Kirpides runs this Genomes Online database that helps us determine which genomes have been sequenced or are in progress. Phil Hugenholz helps work with this Green Genes database, which is a database of ribosomal RNA sequences and phylogenies of bacteria and archaea that allows us to look at where those genomes fit relative to the phylogeny of bacteria and archaea. And we basically merged these two databases, or Phil and Nikos and Danying Wu in my group merged these two databases and looked for branches that had cultured representatives but no genomes available. We identified 200 of these and selected 107 or so as our top priority for sequencing. And we basically did this in two ways. We sampled as diverse as we could from across the tree, and we zoomed in within the Actinobacterium phylum to sample that phylum in a little more depth than had been done even previously. Even with all the genomes that had been sequenced, it was not sampled very well. So the list of the organisms being sequenced is available on the JGI homepage. There's a GEBA sequencing list page, and they're also in Genomes Online. And then what I'm going to tell you for the rest of the time is sort of how the project has worked out and what we've been doing in the project. So one thing that's really important, in particular for someone who's actually an adjunct scientist at JGI, and I spend too much of my time at UC Davis probably, but trying to spend more time at JGI, has been this amazing project management team that many of you have interacted with in the course of doing your projects through JGI that has coordinated this project. So they've developed a whole pipeline for bringing in lists of organisms, working with the culture collections, getting the DNA, QCing the DNA, tracking all the organisms, sending me and everybody else on the team updates, and has been instrumental in getting this project to work. Without that project management team, there's no way we could have handled this scale up in terms of the sequencing within the course of one project. A second really important part of this project has been this partnership that we've developed with this culture collection, the DSMZ. And this was done basically because we realized that getting DNA from all these organisms was going to be the bottleneck in this project. And the solution for us was not to grow all the organisms ourselves, but was to find other people to grow the organisms for us. And fortunately, the DSMZ culture collection offered to do this for free, with no charge for what probably cost them $1,000 or so or more for each organism that they were growing. And ATCC, we got a couple of DNA samples from them for a fee. JGI is in discussions now with some other culture collections such as PCC to fill in some other groups that we missed in DSMZ. But this partnership with the DSMZ has been instrumental in getting some of these weird thermophilic, anaerobic, hard to grow organisms cultured and get DNA in-house. They even do things like this, like write up whole DNA QC figures and email them to us and build a database of all the information. They've been amazing. So partnering with a culture collection is great for a lot of reasons, including getting the DNA, but then the strains are available to everybody in the world without us having to provide them. So the current status of the project is that over 100 genomes are in some phase of progress in the course of this project. We sort of selected the first 56 genomes that have come through the pipeline to be the focus of analysis for whether or not this is a beneficial thing to do. 34 of those are finished. 55 of the 56 have been submitted to GenBank. The data is available on the JGI FTP site and in a special IMG GEBA page within IMG. And the data is open and available for anybody to use. This is the homepage. If you just do a Google search for IMG GEBA, you can find the page, I think, through there. And you can get the link from me if it doesn't show up in Google. There's also been a side project that has spun off of this, run by Cheryl Kerfeld at JGI, which has been to try and get various undergraduate institutions in particular to adopt these organisms for people to use in the course of a bioinformatics class or a microbiology class or a genomics class, and to look at the annotation and to use them in some way. And this is a very exciting project that allows people to get their hands on really recent data and participate in the course of this project. So the rest of what I'm going to talk about is analyzing these genomes and trying to figure out what the value that comes from them is. So all genomes, we think, have some value for a variety of purposes. What is the benefit in particular of this tree-guided sequencing over, say, random sampling of genomes? So we outlined in the original sort of vision of this project these 5 areas that we thought should be benefited from having better phylogenetic sampling of bacterial and archaeal genomes. And that's the list of them here, and I'm going to go through each of these quickly to show you what we've found for some of these. So before I do that, there was one thing that we had to do, which was we used the ribosome RNA tree as a guide for selecting genomes. But there have been many studies, I've even authored some of them, that say that the ribosome RNA tree is not perfect for predicting the phylogeny of species, that genome-based phylogenies are somewhat better than the ribosome RNA phylogeny for a variety of purposes. So we wanted to know, if we selected based upon the ribosome RNA tree, did we, of those 100 or so genomes, how many of them were actually phylogenetically novel? Did it just completely mislead us, or is it just a subtle difference? And basically every way we looked at this in terms of the genome phylogeny, by doing phylogenetic trees of the sort of housekeeping genes in the genomes, these genomes are truly phylogenetically novel. The exact branching pattern in the tree may be slightly different than the ribosome RNA tree, but selecting organisms by the ribosome RNA tree identifies organisms that are truly phylogenetically novel in the bacterial and archaeal history. So in terms of these 5 areas, one of the things that we thought was most important was to use the genomes to improve our ability to discover new functions or new types of genes. And the premise behind this is simple. If you have an organism that's been evolving separately from other organisms for a long period of time, it has the potential to invent new processes, new pathways, new networks. The one caveat with this in bacteria and archaea is that, well, lateral gene transfer does happen. Some people think it is all of the signal of bacterial evolution. And so the question is, if we use the sort of core of the genome, the ribosomal RNA tree or these housekeeping genes, to identify novel branches, well, if there's been a lot of lateral gene transfer, that might not be of any value in predicting the novelty of functions within those genomes. So we basically tested this in a variety of ways. I'm going to show you one of them, which was to take all the genomes, a variety of genome data sets, and ask, how many protein families do we see in these genome data sets? As we add more and more genomes in these data sets, what is the rate of recovery of new protein families? This is a rarefaction curve, basically, for protein families. And Dunying Mu and my group did this. So for example, if you do this with the Streptococcus genomes, which were the basis for this pangenome hypothesis about the presence of lots of novelty within new strains within a genome, you see some increase in, I don't know if this is even working, some increase in the number of protein families as you add more and more genomes, but not a big increase. If you do this for a family, the Enterobacteriaceae, you see a higher rate of increase of recovery of new protein families per genome that you add. If you do this for a phylum, you see an even higher rate of increase of new protein families per genome that you add. And if you do this for the genome sequenced in these first 56 genomes, we see an even higher rate of increase of novel protein families relative to the protein families within one of the most functionally diverse phyla that's available in bacteria. So we think that this sort of says that, at least in terms of sequence-based protein families, we discover more of the new ones by sequencing phylogenetically diverse genomes than by sequencing other types of genomes. So this is not due to the genome size, by the way, in these genomes. If you correct for the number of proteins, if you look at this in terms of the number of proteins per genome, they're consistent across all these different groups. So this is due to true phylogenetic novelty of the protein families within these genomes. So one question you might ask is, are there structurally novel proteins? Are we just detecting sort of new subfamilies within families by our cutoff threshold for discovery of protein families? And it certainly looks like within these GEBA genomes there are structurally novel proteins that have no good matches to crystal structures that are available in the databases. And Patrick de Hasselaer did some of this structural analysis, and Cheryl Kerfeld has gotten a selection of about 350, 370 of these to now be crystallized by the Protein Structure Initiative as representatives of novel structural folds or structural families of proteins. So in terms of novelty, another thing that's important is the diversity within known families of proteins, not just new families, but diversity within known families. And we've asked basically every expert we can think of that focuses on a particular process to look at this. I'm going to give you one example. So Ian Paulson has looked at the transporter profiles, as has Ian Anderson, who's at JGI, and both of them came to sort of the same conclusion, that was that there's certainly some novelty within these genomes. And for example, both of them highlighted the same thing, so I'm highlighting it here. This one organism, this organism that was isolated from a termite gut, has twice the number of sugar PTS transporters of any of the previously 1,000 sequenced genomes. So this is a functional novelty within a class of proteins that we knew about, but the diversity within that family is more than has been seen in any previously sequenced genome. So in terms of novelty, another thing that is of interest, especially to people working on metagenomics, is the novelty within a presence of particular protein families across the tree of life. So the best example of this that has come up has been the proteorhodopsin, which nobody thought to look for in bacteria because it had only been seen in archaea previously, and then when Ed DeLong found it in these bacteria from the ocean, it completely changed our perspective on where rhodopsin-like proteins should be found. Victor Koonin at JGI did an analysis searching for these types of proteins by comparing the GEBA genomes to, for example, proteins that had previously only been seen in eukaryotes. And he found a list of a bunch of these, and one of them in particular is really striking. This is the first true actin homologue to be found in any bacterial genome. So bacteria have an actin-like protein, FTS-Z, that works in many ways like actin, but it's not a close relative of actin. Oh, sorry, not MRE FTSE is the tubulin homolog. I forget what the actin-like protein in bacteria is. But this is an actin homolog in bacteria, very closely related to eukaryotic actins. You can even thread it against the known structure of actin from different organisms. It's actually, I did this tree last night because I wanted to show something phylogenetic to everybody. You know, evolution is everything. And it's actually interesting. This actin-like protein is not in the same family as the true actins, but it's actually in a family of these things called actin-related proteins, ARPA. We don't know what this does. Mitch Singer at UC Davis has shown that you can detect it being expressed in the cells of this organism, but we don't know what its function is. My guess is it's some type of oncogene-like function, that it's trying to disrupt actin polymerization in some eukaryote through some sort of dominant negative property, but we don't have any evidence for what it's exactly doing. But it's, 100 genomes have been sequenced, and many have been partially sequenced. There's not a single one with an actin-related protein like this until this organism. So in terms of the other possible things that can come from this project, another one that we think is going to be very beneficial is the annotation of other genomes. So not studying these genomes themselves, but using them to help annotate other organisms like, I forget who yesterday said instead of model organisms we should call them reference organisms, like Bacillus subtilis, a reference organism, or some other important organisms that you might be interested in. So increased phylogenetic coverage should help with this. This has been shown in animal sequencing, for example, that it does help. So does it help with bacteria and archaeal sequencing? We think it does. So basically we've looked at 3 different ways to test this. You can convert completely hypothetical, sort of unique proteins into conserved hypotheticals by having a match to these genomes that gives you better evidence that they might actually be real as opposed to spurious. You can link distantly related members of protein families that previously you might have thought they were in different protein families, but now you find them to be in the same distantly related protein family. And you can use these genomes to improve non-homology functional prediction methods, which include things like phylogenetic profiling, the Rosetta Stone method of predicting functions. These genomes help with that. I'm just going to sort of quickly show, well, you probably can't see the exact data, but when Nikos Kyripides and his group looked at how these genomes affected the prediction of function or these linking of gene families, they found that they improved the linking across protein families more than you would expect for a random genome. They improved the fusion-based predictions of functions a little bit more than you expect for a random genome. And similarly for these Rosetta Stone functional predictions, they seem to improve it more than you would expect for a random genome. So it looks like adding better sampling across the tree improves your ability to use these non-homology functional prediction methods to annotate other genomes. So, metagenomics is being done in every possible place you can imagine, and unless you're working on the human microbiome or some good model system, you're probably not going to have a collection of reference genomes to help you analyze your metagenomic sample very well. We think that sampling across the tree of bacteria and archaea will provide sort of a phylogenetic scaffold for all those other projects that don't have closely related genomes to help them scaffold their metagenomic data. And so what we've tested so far is basically how do these genomes help with annotating metagenomic data. So, short sequence reads, you frequently don't even get blast matches to even have any ideas to predict the functions of those genes. You can use these GEBA genomes to help match up short fragments to a protein family. And Nikos' group has shown that this definitely improves with adding these GEBA genomes. If you look at compositional-based binning of sequences, which is one of the approaches to bin data, we see a moderate, it's not incredibly large, but a moderate improvement in compositional-based binning by adding these GEBA genomes. And we see my slides going away. I'm going to slowly open that back up. I don't know what happened. So in addition to looking at this compositional-based approach, we've also seen a phylogenetic approach to binning has improved slightly by adding these GEBA genomes. So we haven't revolutionized all metagenomics by doing these first 56 genomes. We think that's because the tree has not been sampled incredibly well still hang on a second here. Sorry about that. We think that the metagenomic improvement has not been great because the diversity of bacteria and archaea is immense. And we just haven't scratched the surface of it by these first 56 genomes in order to really help metagenomic analyses in a large way. So in terms of phylogeny, we think that, again, sampling genomes from across the tree should help, and this is not from the GEBA project, but from prior work that we did with Jonathan Badger and Naomi Ward, which showed that, for example, if you look at, this is what I was mentioning before, the 16S tree of this organism, Hyphomonas, gives a misleading classification of this organism. It's sort of deep branches in the phylogenetic tree that the 16S is different from everything else in the genome. So we've looked at the phylogeny of all the genomes that are available right now, including the GEBA genomes. This is using a method called Amphora that is an automated phylogenetic tree building method that Martin Wu in my lab developed. And if you zoom in within the Actinobacteria, for example, in red are the GEBA genomes, in black are all the other genomes that were available. If you look within this tree, we definitely see differences between the phylogeny that we have versus the phylogeny that was available from ribosomal RNA previously. And we're in the process of looking at how robust these differences are and whether they're going to cause us to reclassify some of these groups of Actinobacteria. So we think that these 5 areas that we proposed are going to be benefited by having this phylogenetic sampling all are being benefited. Maybe at different levels, but they all are getting improved by this phylogenetic sampling from across the tree. So the question is what to do after this pilot. So if you look at, for example, the phylogenetic diversity of bacteria and archaea, and you ask, in all the genomes that have been sequenced so far, what is the total branch length in the phylogenetic tree of those genomes? And you rank them, in essence, in order by the most diverse over here, and then you add in the least and least diverse. So, you add in 75 E. coli genomes, they don't add a lot of phylogenetic diversity to the tree. If we then add the GEBA genomes to this plot, each one that we've been doing adds a lot of phylogenetic diversity to the total tree here. So we're still sampling lots of phylogenetic diversity in the bacterial and archaeal world. If we actually zoom out and look at all the bacteria and archaea ribosomal RNAs in the database, we have a long way to go to fully sample the bacterial and archaeal tree. But if we look at just the cultured representatives, we actually think it's very tractable to get to a point where we have, say, half of the phylogenetic diversity of those cultured representatives sampled by maybe 1,000 more genomes or so. We don't know the exact numbers yet, but it's something on the order of 1,000 more genomes to cover half of the phylogenetic diversity in all the cultured species. So we think that this is actually tractable with the new sequencing methods to really sample the bacterial and archaeal tree very well. Of course, we've only focused so far on cultured species. Eventually, we're going to have to fill in the branches for all the uncultured lineages, and I think you'll hear in other things here and in lots of the posters how people are trying to approach that. The same trend is seen in viruses. So the phylogenetic sampling of viruses has not been great. The phylogenetic sampling of microbial eukaryotes has not been great. The phylogenetic sampling, in fact, of almost any group has not been great if you take the perspective of zooming in beyond sort of the family or order level of most of the groups. So fungi, plants, lots of these things have not been sampled very well. There are projects, many of them at JGI, in progress to improve the sampling of those groups also. And of course, just sequencing genomes is not enough. One of the reasons to have this Adopt a Microbe campaign is to get people to start to look at the genomes and work on those organisms. You need experiments from across the tree in order to place a lot of this information in perspective. So we need people to start working with these organisms. That's one reason to release the data immediately, to get things from culture collections so anybody can get the organisms. And in the long run, my dream is, I'm a birder. I work on microbes, but I'm obsessed with birds. But what I want to have is, like we have for a field guide to the birds, is a field guide to the microbes, where we have the phylogeny, the biogeography, and the diversity all there in one place for us to look up and then interpret all the experimental studies. And then we'll have a happy tree of life.
Thanks, John.
So Jonathan, you showed us a slide right near the end that had about 3 billion years worth of evolution. If you could go back to that, it's all the whole genome-based tree. And I wonder if you could comment on, surely that, yeah, there you go. Surely that will differ from the 16S-based tree, no doubt.
Yes.
Anyone would have predicted that, as you know. But if you were to use an MLST approach, would it be that different?
If you were to use an MLST approach instead of sequencing the genomes.
Instead of the entire genomes from each of those, I mean, most of us don't have access to the data you have access to, but we maybe could sequence 5 genes from 100 things.
Yeah, so the question is basically, using the whole genome, does that give you a different answer than sampling, say, a subset of these housekeeping genes? We don't know that answer, actually. We're looking at it right now for another project that I'm working on, which is trying to say, if you have the whole genome tree, and then build phylogenies of all the individual genes, how many of them agree with the whole genome tree, and how different are they from the whole genome tree? So what we did for this is a basically bootstrapping the whole tree. This is a concatenated alignment of 31 housekeeping genes from across the bacteria. It's probably not the best way to do the phylogeny, actually. Everybody in the phylogenetic community seems to think that analyzing each gene separately and then combining the results is a better method. So that's what we're looking at now. But we don't know how many you need and which ones you need in order to do this. So yeah, I think that that's a great question. I think actually, you know, with the cost of sequencing going down, hiring a technician to do PCR is more expensive than sequencing the whole genome. So we can do, yeah. So I think that, you know, 2x coverage with 454 is probably going to be able to do this.
Somebody has to pay the 454 technician.
Yeah.
So John, can you go forward 2 slides? Keep going. One more. Okay, let's talk protein space. So you added a jump from blue to red, which is a jump of, let's call it, generously 2%. 5%?
Yeah, I mean, it's different number of genomes. So this is number of genomes on this axis and the total branch length in the tree on the y-axis.
So you added a tiny increment. You added, I think you said, 170 potential new protein families.
Yeah, 1,000.
About 1,700, right, out of 180,000 total. If you extrapolate that to the whole gray zone, well, is your opinion that we have not even
so we haven't done that yet, but basically what you're asking is sort of to do a equivalent to like a Chow diversity estimate for protein families. We have not done that yet. Other people, so Shibu Youssef in the GOS paper where they looked at the protein family space added by the GOS metagenome data, they looked at that a little bit, but they didn't try and estimate the point of the end point completely. So we have not done that, but it's very easy to do it from this data set that we have. Just, it's a great idea.
John, you are a believer in trees and phylogenies. Was there anything that you were surprised about? Were you surprised that it wasn't more informative about the metagenomic data?
Yeah, I think that, I mean, the question is, are there any things we're surprised about? I think that there are 2 things that I was actually surprised about significantly when we did this analysis. Yes, that the metagenomic metrics that we used did not improve more than we expected. What we've done so far with the metagenomic data has not been comprehensive. So what we did was we selected particular metagenomic data sets, individual ones, and then compared the results with and without the GEBA genomes in terms of analyzing those data sets. We tried not to rig the process completely by picking data sets we knew would benefit from these genomes that we were sequencing. So I think what would be better is to actually sample all metagenomic data sets and really assess this in a comprehensive manner. And then I think in retrospect, I'm not as surprised by looking at the total phylogenetic diversity. But we thought, for example, that, you know, the first representative of an order, many first representatives of orders would greatly improve Megan or phylogenetic-based binning of the metagenomic data. And it did not improve it as much for the data sets that we looked at. And I don't know why yet, so we're still looking at that detail. The other thing that, I mean, as an evolutionary phylogenetic person, I should have been more ready for was these plots of the discovery of protein families. I did not expect to see as strong a phylogenetic signal in those plots as we found. I was, you know, because these genomes are from across the sort of ecological diversity too. So if you sample a thermophile versus a sulfur metabolizing mesophile, do you discover lots of new protein families there? And you do. So I didn't expect, when you just sort of sample from across the tree, so many more protein families to be recovered as you sample further and further across the tree. So those sort of two things are the big, we really didn't expect that. And many of the other things are, you know, they've already been sort of shown for other projects, like the animal projects, that annotation should improve. And yes, it does improve. That's not that surprising. So those are sort of the two.
So this, quick question out of curiosity. So we know that, so you're probably generating these protein families based on blast sequence analysis, right? And so there's a shuffling of domains. You actually are going to get sort of a skewed signal in terms of how many new families you're finding. So I'm just curious, if you drill down, do you actually find differences at the domain? Are you finding new domains, for instance, structural domains?
Yeah, so the way we did the protein families was, it was this MCL clustering based upon blast results, so it's exactly what you're saying. And you hope that it teases apart some of these multi-domain families, but it doesn't do a perfect job with that. So that's why when we looked at those, so 1,000 of the families that we found had no blast match of any kind to anything in GenBank. So those families that it calls as novel families, it's not that they have some weird domain structure. They're completely novel in terms of sequence. Those are the ones that are being fed into the Structural Genomics Initiative. But if you look at the other ones, we haven't yet completely teased apart when you add more genomes, the families grow and shrink in various ways by exactly this sort of transitive linking that you're talking about. We haven't analyzed that in detail to see what happens with multi-domain proteins getting merged together into families versus not.
So the following question to that is, within the new protein families that you find, how many sort of these subsequences that potentially represent domains are conserved? Have you done that kind of analysis?
Within those families. So
new families.
I didn't show that, but we have looked at the number of representatives of those families within this data set. But we haven't looked at whether or not they have conserved domain structure. They have no sequence similarity to anything that's been found in any database.
Right.
So it's all within those protein families, but we haven't looked at structure within them.
Yeah, I'm sorry, we have to take a break. We have 15 minutes. We'll reconvene here at 10:30.